lymphoid tissue. We'll be talking about some of the organs, primary lymphoid organs, secondary lymphoid organs, which are concerned with mounting the immune response, exposure to the antigens like there can be bacteria or protozoa. To tackle those immune problems, we have some of the organs, and let us look at a diagram to discuss them now. In this diagram, we can discuss the lymphoid organs like we have primary lymphoid organs, 2 examples given here, and then we can also talk about secondary lymphoid organs with few examples there. But first, we are starting with primary lymphoid organs. These primary lymphoid organs are called so because they are the one who will be having the stem cells. And if you do not have the stem cells, then it is secondary lymphoid organs. So this is the basic difference between the 2 types of lymphoid organs. And then we have to mention it is the stem cells which will be forming the lymphocytes, and the process is called as lymphopoiesis. And once we have the lymphocyte formed in the primary lymphoid organs, then they will be transferring it into the blood circulation and can move on to secondary lymphoid organs, which are basically involved in mounting the immune response, but they do not have the stem cells. They have already formed lymphocytes. Now, coming to the primary lymphoid organs, we will see the first name to be taken is red bone marrow, because that is where we have the formation of lymphocytes. And from the red bone marrow, the lymphocytes are moving towards the thymus. In fact, the stem cells which are there in red bone marrow, they migrate towards the thymus, and thymus also has the stem cells. That is why it is considered as primary lymphoid organ. Now, the thymus, which is having the lymphocytes, they are called as T lymphocytes. And T lymphocytes, they are basically for cell-mediated immunity. And this immunity is against the organisms like the bacteria or protozoa, and this is basically the first line of defense. Now, this is about the T lymphocytes. What about the B lymphocytes? We'll find that the red bone marrow having some stem cells, they were changing into lymphocytes, and those lymphocytes you can call as B lymphocytes. So, red bone marrow is having the B lymphocytes. What is their function? They will be actually converting themselves into plasma cells, and it is the plasma cells which will be making the antibodies. Here, when you are talking about the antibody-mediated immunity, this is having another name. We also call it as humoral immunity. And humoral immunity is a bit delayed if you are comparing it with the cell-mediated immunity. So here, as you notice, the stem cells which were there in the bone marrow converting themselves into the lymphocytes called as the B lymphocytes themselves convert into plasma cells, later forming antibodies, and this is humoral immunity. Let us discuss one clinical situation here. It is called as the D-George syndrome. So which of the 2 are involved? If we are talking about the D-George syndrome, then we see the problem is with the thymus. The thymus is absent, and if the thymus is absent, you cannot train the stem cells as T lymphocytes, so T lymphocytes will be absent. It means that the cell-mediated immunity is absent, and that is why these babies who are suffering from the D-George syndrome, they will be dying because of the severe infections, bacterial infections, protozoal infections. Now, let us move on to the details of secondary lymphoid organs. They can be capsulated or not capsulated. Like you will see under the capsulated format, we can take the lymph nodes. One more example of capsulated lymphoid organ in the secondary lymphoid organ category, you can consider the spleen as well. So lymph node and spleen are capsulated. They will have a capsule made up of the collagen fibers. And what about the non-encapsulated? The tonsils are non-capsulated. Tonsils are under the category of mouth mucosa-associated lymphocyte tissue, and they do not have a capsule. Then what are they lined by? They'll be lined by the mucosal epithelium. Another example of non-encapsulated mouth is here, the pear patches. You will find in the terminal ileum, we have got another mouth mucosa-associated lymphocyte tissue, which is known as the pear patches, aggregation of the lymphoid follicles. And even here, the lining is that of the mucosal epithelium. Yet another example of mouth can be seen in the appendix as well, the mucosa-associated lymphocyte tissue. We'll be looking at some histological pictures describing the details of vermiform appendix or these secondary lymphoid organs. Plus, we have to look at the histological picture of thymus as well. The discussion goes according to the the type of questions we encounter in the exams. What we are looking at is the histological picture of lymph node, thymus, and here is the spleen. All the three are capsulated lymphoid organs. Then you will notice that in case of a lymph node and a thymus will have the peripheral cortex and inner medulla. Peripheral cortex and inner medulla. But this cortex and medulla is not found in the spleen. In the spleen we'll have red pulp and the white pulp. Now let us look at some more details of these lymphoid organs. We are starting with the detail of the lymph node here, and we have magnified the diagram. As you mentioned, these are all encapsulated organs, and the capsule which we are talking about is actually made up of the collagen fibers covering the organ. One of the identifying feature of a lymph node is just deep to the capsule, we will have large subcapsular sinus. And what is filled up in the subcapsular sinus? It is the lymph. The lymph which is being brought by afferent lymphatic vessel, as is evident. This lymph is going to be filled up into the subcapsular sinus, and from there it is trickling down from the peripheral cortex towards the inner medulla. So there is a peripheral cortex, inner is medulla, and from there the lymph will be trickling inside the efferent lymphatic vessel, the outgoing lymphatic vessel from the lymph node. So you can show the lymph flow as it is coming from the afferent lymphatics, enters the subcapsular sinus, passing through the cortex, going into the medulla, and then moving into the efferent lymphatic vessel. Now, as we are marking, in the periphery we have got the cortex as evident here, and then as you move deeper, this area is the region of the medulla. You can mark the inner central portion, that will be the region of the medulla. Then we have to mention one intermediate region as well, which is called as paracortex. So this will be the region of the paracortex. We have got the outer cortex, then we have paracortex, and then we have the inner medulla. Now, how do you identify the peripheral cortex? There we have got the lymphatic nodules, or what is called as lymphoid follicles. And this is collection of B lymphocytes. In these lymphatic nodules, there is a central portion which is called as the germinal center. And in the germinal center, we have the B lymphocytes undergoing hypermutation. Basically, there is exposure and the B lymphocytes are undergoing differentiation, getting ready to mount the immune response accordingly as the exposure to antigen is there. So in the cortex, we are having numerous lymphoid follicles, having a lot of B lymphocytes which are being trained in the germinal center, then they move towards the periphery of the lymphatic nodule. What is there in the paracortex? In the paracortex, we have thymus-dependent T lymphocyte zone. So outside we have B lymphocytes, and deeper in the paracortex, we are having T lymphocytes, or this is thymus-dependent lymphocyte region. And what do you have in the medulla? In the medulla, we have the medullary cords and medullary sinus. Sinus will be filled with the lymph which we were talking about, and they are surrounded by medullary cords. Then what is there in the medullary cords? In the medullary cords, we have got some cells. You can remember a mnemonic LMP. And what is the full form of LMP cells? The lymphocytes. M are the macrophages, and P is for the plasma cells, the modified B lymphocytes. Now, we also will find that towards the hilum of the lymph node, there are blood vessels coming in. The lymphatic vessels are going out and blood vessels are coming in. That is the artery. Artery will come, vein will be going out. Now, as the arteries will enter, they'll change into arteriole, capillaries, and then we'll have the venules, collecting the venous blood and sending it back into the vein. The venules here are specific in features called as high endothelial venules. And these high endothelial venules, they have the capacity to send out the lymphocytes from the blood circulation directly into the lymph node. And that is a typical feature here. In the lymph node, having this high endothelial venules, transferring the lymphocytes from the circulation into the lymph node. Now, let us move on to the detail of the thymus as well. Thymus will be having multiple lobules, and there will be multiple cortex in the periphery, medulla on the interior. So, as you will show, in the periphery, we are having this cortex as marked here. And when we look inside, this region is the medulla. In the medulla, we are also having a specific feature, Hazel's corpuscles, which are made up of epithelio-reticular cells which are degenerating, and we'll be finding them in some histological specimen as well. So thymus will be having multiple lobules with the outer cortex, inner medulla, as is evident here as well, outer cortex and inner medulla. Now before we look at the further details of the thymus, let us briefly look over the structure detail of the spleen as well. As we mentioned, these three are capsulated lymphoid organs including the spleen, and the capsule is basically the collagen fibers. As you can see, the capsule is sending some partitions inside the substance of the spleen, and we are calling it as the trabeculae. It is kind of a partition. But in the spleen, we are not having the outer cortex, inner medulla. There we have got red pulp and the white pulp, and most of the area is occupied by the red pulp. That is about 70 to 90% of the constituents, and the remaining is white pulp. That means 10 to 30%. White pulp means you'll have some lymphoid nodules. As you can see, they are scattered, and surrounding these lymphoid nodules, this white pulp, all of the rest of the area here is the red pulp. Now, in the white pulp, the lymphatic nodules, we'll find they are having mostly the B lymphocytes, and these B lymphocytes, they are present in the inner germinal center and in the peripheral area as well. In the germinal center, the B lymphocytes, they are undergoing hypermutation. Exposure to the antigen leads to their differentiation, and as they get matured, they move on towards the periphery. Now, if it is all B lymphocytes, then where are the T lymphocytes? For that, you have to understand that the lymphoid follicles will have something called as central arterioles. Now, maybe they are called central arterioles, but they are not in the center. They are in fact eccentric, means off center. And the central arterioles, which are in fact eccentric in the location, they are surrounded by some T lymphocytes. And those T lymphocytes, which you can show, they are the one which are called as PALS, peri-arteriole lymphatic sheath. So they are making a lymphatic sheath around the arteriole, PALS, and they are the T lymphocytes. So around the central arteriole, you have T lymphocyte, whereas most of the lymphoid follicle is B lymphocyte. They are all white blood cells, and this is the white pulp. And white pulp is quite limited. It is surrounded by the red pulp, which is basically having more of RBCs, hence the name red pulp. But be careful, not only you will find RBCs here, but you can have presence of WBCs as well. Then why are you calling it as red pulp? See, what is the ratio of RBCs and WBCs in the blood? Because the spleen has the spillage of blood, and in the red pulp, if you look at the proportion, RBCs are 5 million per unit of blood, whereas WBCs are 5,000 per unit of blood. At one place 5 million, other place 5,000. If you mix them, obviously which one has the large number? So despite the fact that in the red pulp you have RBCs plus WBCs, but since it is more of RBCs, it appears red in color and hence called red pulp. In the red pulp, you'll find the splenic sinusoids surrounded by splenic cords as well. So in the red pulp, we are having the splenic sinusoids plus the splenic cords as well. And what is there in the splenic cords? Like if you remember what you found in the medullary cord of the lymph node, like in the medullary cord of the lymph node, you were having some LMP cells. Even here in the splenic cords, you'll have the LMP cell. And what is the full form of LMP? L for lymphocyte, M for macrophages, and P is the plasma cell, modified B lymphocyte. Now we are going to look at more specific details. Just now we have had just an overview of these three organs. Let us start by drawing a diagram of lymph node now. As we mentioned, the lymph node is having a capsule which itself is made up of collagen fibers, and the framework of lymph node or any lymphoid organ except thymus, we are having the reticular connective tissue. If you remember, it is the reticular connective tissue which is type 3 collagen fibers forming the framework of the lymphoid organs. Now, just deep to this capsule is a large subcapsular sinus, and that large subcapsular sinus will be filled with the lymph, which is brought by the afferent lymphatic vessels, and this lymph is moving towards the inner medulla. So basically we are having one outer cortex, cortex and inner medulla. There is one paracortex also. Now, how do you identify the cortex? In the cortex region, we are having the lymphatic nodules, which are occupied by the B lymphocytes. So here, as you look at the lymphatic nodules, which are basically B lymphocytes, there will be some germinal centers as well. So there are some germinal centers to the interior. They are having lightly stained as compared with the periphery, which is darkly stained. And it is the germinal center where we have the B lymphocytes undergoing hypermutation, differentiation, identifying the antigens, and preparing themselves to become the plasma cells. And these plasma cells then move from the cortical region towards the medullary region. So what will be there in the medulla? These modified B lymphocyte, the plasma cells, and some more cells, which we'll be talking about. But you have discussed the cortex having B lymphocyte. Where are the T lymphocytes? As you come deeper, there is a paracortex. There you will find the thymus dependent region, the T lymphocyte region. So here you can mention in the paracortex, slightly deeper, we have the thymus dependent region. The T lymphocytes are found here. And as you move still deeper towards the medulla, you will have the medullary sinuses filled with the lymph, as we have mentioned. And there are some medullary cords also. In the medullary cords, you are having the LMP cells. So what is the full form of these LMP cells in the medullary cords? L, the lymphocytes. And what about the M, the macrophages? And P, you already know, the plasma cells. So there are some cells in the deeper region medulla. Medullary sinus is filled with the lymph, which came from the afferent lymphatic vessels passing through the subcapsular sinus, and the cortex, paracortex reaches the medulla. Then goes towards the hilum of the lymph node, and from there will be some efferent lymphatic vessels taking lymph outside. So this is about the basic structure of a lymph node. Let us look at this in a histological slide. In this diagram, you'll find there are a lot of blue dots, and that is a typical appearance of any lymphoid organ, may it be the lymph node or the thymus or the spleen. So as you magnify one of the blue dots, they are basically the lymphocytes. And lymphocytes are identified by having a very large nucleus almost occupying entire of the cell with a peripheral thin rim of the cytoplasm. And because of this arrangement, the lymphocytes are appearing as blue dots, thousands and thousands of blue dots. Now, this is a lymph node and it is having this outer capsule. It is made up of collagen fibers. As we mentioned, this is the capsule region, and this capsule is actually sending some septa inside called as the trabeculae and causing septation of the lymph node. So these are the trabeculae and they are moving from the capsule towards the cortex and paracortex. So how do you identify the cortex? First of all, it is in the periphery. Deep to that will be the paracortex, and still deeper if you move, then there will be the medulla region. So as you see, this is the cortex region, and then you have the paracortex, and a part of the diagram here is showing the medullary region of the lymph node. Before you talk about the features of the medulla, tell us about the cortex. In the cortex, we are having lymphoid follicles or lymphatic nodules having B lymphocytes, and the lymphocytes which are towards the germinal center, lightly stained region, as you can make out. This is the lightly stained region, the germinal center, whereas this is the outer peripheral region. In the germinal center, we are having the B lymphocytes which are undergoing exposure to antigen, hypermutation, differentiation, getting ready for mounting the immune response, and then they move towards the periphery, which is slightly darker region in appearance. And one specific feature which we have mentioned, in the lymph node, there is a large subcapsular sinus which is filled with the lymph as brought by the afferent lymphatic vessels. So there is a gap between the outer capsule and the inner cortex region, and this is one identifying feature of the lymph node. As you mentioned, this is the large subcapsular sinus. Once you see that, your identification goes in favor of lymph node. So as we understand, there will be some afferent lymphatic vessels bringing the lymph here, and then lymph is moving through the cortex, paracortex towards the medulla. Now, what do you find in the paracortex? Paracortex, they have no B lymphocytes. That is the thymus dependent region, and there where you will have T lymphocyte. So in the paracortex, we are having T lymphocyte, the thymus dependent region. What about the medulla? In the medulla, we are having the medullary sinuses filled with the lymph, and along with the medullary sinus, you have the medullary cords. And in the medullary cords, we know there are LMP cells, the lymphocytes, which could be B or T lymphocyte, and then macrophages, and the plasma cells which are activated B lymphocyte producing antibodies. So basically, we are trying to mention this region is concerned with the humoral immunity, that is the cortex, whereas the paracortex is related with cell-mediated immunity, because here you are having T-dependent region, thymus-dependent lymphocytes, and then when it comes to medulla region, you have T lymphocyte as well as B lymphocytes, along with the lymph. The lymph itself, which is brought from outside, is bringing some of the antigens, antigen-presenting cells, and they keep modifying the function of B lymphocyte, T lymphocyte, macrophages, all that in the lymph node. Thymus is a multilobulated structure, and we'll find there are multiple lobules. And each of this lobule will be having the peripheral cortex and inner medulla. So you'll find that this kind of arrangement, in the periphery there will be cortex, and in the center there will be medulla. And in the medulla we'll be having a very typical finding, Hazel's corpuscles, which are basically degenerating epithelial reticular cells, which we'll be finding in one histological picture as well. The function is unknown for Hazel's corpuscle, but some authors will say they are giving us what is called as the cytokines for the purpose of providing immunity. Now, thymus is one organ where you do not have reticular connective tissue. We have mentioned reticular fibers are basically type 3 collagen fibers, and they are missing from the thymus. Thymus doesn't have a framework provided by these reticular fibers. Then how the lymphocytes are staying inside the thymus? The framework is actually provided by the thymic reticulocytes. So there are some cells which are called as the reticulocytes in the thymus, and it is the framework being provided by them in which the lymphocytes will be staying. And basically, it is the thymus, so it will have T lymphocytes arranged in the lightly stained medulla, which is inner, and darkly stained cortex, which is outer. So cortex is darker stained and medulla is light stained. Let us look at some diagrams now, giving us some more details in pictorial form. So as we can see, there are multiple lobules in the thymus. Thymus itself is a bilobed structure, right lobe, left lobe from the front view. And as you take a section of one of the lobule, you will find each lobule is having multiple cortex and the medulla. Cortex is towards the periphery, as you can notice here, and is darkly stained, whereas medulla is towards the center and lightly stained. And these blue dots, what you find, they are all the lymphocytes with a very large nucleus, thin peripheral rim of cytoplasm. So just by looking at these multiple dots, we can see it is a slide of the lymphoid organ. And we still magnify the region. As you do so, here you can find the outer cortex and the inner medulla. So, in the cortex, you will find there is no type 3 collagen fiber or reticular fibers. What you have is the reticulocytes, the reticular epithelial cells. So, these reticulocytes, they provide the framework where you can hold the lymphocytes, the T lymphocytes, because this is thymus. Here the stem cells are differentiated into T lymphocyte, cell-mediated immunity to fight the bacteria, the protozoa, and like any other capsulated organ, the outer capsule sending inside what is called as the partition or the trabecula. Now this is about the cortex, which is darkly stained. What about the medulla? In the medulla, there is a typical feature, the thymic Hazel's corpuscle, and let us magnify the Hazel's corpuscle then, which you can see in the next diagram. Numerous reticulocytes arranged in a concentric fashion, and there is a degenerating center as well. So here is a slide showing the thymic corpuscles and these different cells which you find, the blue dots, they are all thousands and thousands of lymphocytes. So these Hazel's corpuscles, basically they are a collection of reticular epithelial cells arranged in a concentric fashion with a central degenerating eosinophilic matter. And as I mentioned, the function is unknown, but some of the authors mentioning they are providing us cytokines for immunity purpose. A typical feature of thymus is the thymic Hazel's corpuscle. As we understand, spleen is a secondary lymphoid organ. It is an encapsulated organ and it is not having a peripheral cortex and medulla. Instead, it is having red pulp and white pulp. Now, when we say the spleen is having red pulp and the white pulp, what is the percentage, the proportion among the two? For that, we can mention mostly it is red pulp, 70 to 90% portion of the spleen, whereas if it is white pulp, then it is 10 to 30%. Now, when you say white pulp, it means there will be more of WBCs, and WBCs also the lymphocytes, because this is one of the lymphoid organ. And these lymphocytes could be B lymphocytes or T lymphocytes. Now, when you are talking about the white pulp, the WBCs, the lymphocytes, what about the red pulp? In the red pulp, as the name is suggesting, there will be more of RBCs. But of course, it will have some WBCs as well. The count, RBCs are in millions, whereas WBCs are in thousands. So if you mix them, still there will be more of RBCs, hence red pulp. You will notice that under red pulp, there are some WBCs to be described. Now, in the red pulp, let us talk about two components, the sphenic sinusoids filled with the blood, and then there are sphenic cords of Billroth. As you will mention, there you will find some of the WBCs. But first we will be looking at the sphenic sinusoids. So what is there in the sphenic sinusoids? You'll find they are filled with lot of blood and lot of RBCs. Spleen is actually a filter of blood. The blood gets spilled in the spleen and the spleen will clear the blood of any unwanted substance or the constituents or the contents. Like spleen is filtering the old RBCs, they'll be broken down and the constituents sent back to bone marrow. So it is filtering the blood of the old RBCs, only young RBCs can survive. Now these are the sphenic sinusoids and they are covered by sphenic cords of Billroth. As you will show, around them are some cords. These are the cords of Billroth. And what is there in the cords of Billroth? The LMP cells. And what is this LMP cell? L is for the lymphocytes, which could be the B lymphocyte or T lymphocyte. Then M is the macrophages. And the P is the plasma cell, which is activated B lymphocyte to produce the antibodies, humoral immunity. We have seen this even in the medullary region of lymph node. The LMP cells. There it was lymph node medullary cords and here it is the sphenic cords. And these are WBCs. So as I mentioned, though it is called red pulp, but it is having WBCs along with the RBCs. Otherwise, most of the WBCs will be found in the white pulp. Let us look at the two components of white pulp as well, which are having the lymphatic nodules, also called as the lymphoid follicles by some of the authors. And number two, PALS, peri-arterial lymphatic sheath. These are actually the T lymphocytes, whereas if you are talking about the lymphatic nodules, they are having B lymphocytes. So B lymphocytes are present in the lymphatic nodule and T lymphocytes are present in the periphery of lymphatic nodule around the central arteriole as peri-arterial lymphatic sheath. It will become more clear in the diagram. Let us first draw the lymphatic nodule. And as we know, lymphatic nodule, they are multiple in number, having a lot of lymphocytes, the B lymphocytes, as we have mentioned here, having a pale region which is germinal center and a dark region which is in the periphery. In the germinal center, we find the B lymphocytes are undergoing modification. They are exposed to the antigens, the immunocompetency is being given, hypermutation, differentiation, proliferation, and then the trained cells are moving towards the periphery, which is a darker region. Now, as you look at the periphery, you can talk about central arteriole also and peri-arterial lymphatic sheath as well. So what you draw is one arteriole which is coming towards the lymphoid follicle. We call it as central arteriole. Now, despite the fact that it is called central arteriole, it will not reach the center of lymphoid follicle. Say this is the lymphoid follicle with the germinal center. The arteriole stays here outside. That means it is eccentric of center. And surrounding this central arteriole, you have some T lymphocytes as we mentioned here. And these T lymphocytes are making the peri-arteriolar lymphatic sheath. That is the name here. So the point is, in the lymphoid follicles, not at the center towards the eccentric region, there is a central arteriole surrounded by the T lymphocytes making peri-arterial lymphatic sheath. Now, let us say there was a case of septicemia. In the blood, you have a lot of antigens running, the bacteria. So, that blood, which is having the bacteria surrounded by T lymphocyte, as it reaches the spleen, it will spill in the spleen, and these T lymphocytes, they will attack the antigen, the first line of defense. And then in the lymphoid follicle, you have the B lymphocytes changing into plasma cells, forming antibodies, the delayed response, humoral immunity. So when we talk about the lymphoid nodules and the PLS, it is the white pulp because they are all B lymphocyte and T lymphocyte, the white blood cells. And now let us look at a schematic diagram for the same. So this is a schematic diagram of the spleen. We have magnified a portion of it. And then we are looking at the two components of the red pulp and the two components of the white pulp. Let us first focus upon the red pulp. And we are zooming this area. As we have mentioned earlier as well, in the red pulp, we'll be having the splenic cords of Billroth. And talking about the splenic cords of Billroth, they are basically having the framework of reticular fibers, type 3 collagen fibers. As we know, these lymphoid organs will have a framework of reticular fibers. And these reticular fibers are nothing but the type 3 collagen fibers. But along with that, some cells in the cords, the LMP cells, as we have mentioned, lymphocyte, macrophages, and the plasma cells. And we also understand that though these are WBCs, but in the red pulp we can have mostly the RBCs, but even the WBCs have to be included. Like if you are looking inside the splenic sinuses, you'll find they are covered by the splenic cords of Billroth. But what exactly is inside them? Inside them, it is filled with the blood. And in the blood, most of the cells are the RBCs. So these sinuses are filled with blood, in which mostly it is the RBCs, plus of course there will be some WBCs, but you know the proportion. If you compare the two, these are RBCs in millions, whereas WBCs are in thousands. So in effect, there is always more number of RBCs. That is why the name comes as red pulp. So in the red pulp, you have sinuses and the cords of Billroth. What exactly is the function of this red pulp? We have mentioned earlier, spleen is a filter and it can filter some of the unwanted constituents. Like if there are old RBCs, the spleen is going to filter them and the broken down components will be then sent back towards the bone marrow for making new RBCs. So that happens in the sinusoids. The mechanism is elaborate and we are focusing more upon the histological picture here, not the physiology. Now let us zoom out and look at the white pulp, two components as well. And as we do so, we have to understand that in the white pulp, it is mostly the WBCs having the lymphatic nodules, and in the periphery, the central arteriole peri-arteriolar lymphatic sheath. So component 1 and component 2. Let us first focus upon the component 1 and we are zooming this area. So in the white pulp, we are having these lymphatic nodules, one shown here, another one here, the third one here, and they will having germinal centers, the lightly stained area. In the germinal center, we are having the B lymphocytes which are exposed to antigen and they'll be undergoing antigenic stimulation, hypermutation, differentiation, and as they are activated, they move towards the periphery, that is a darker region. Now, this is about the lymphoid follicle or the lymphatic nodule, basically the B lymphocytes. Where do you find the T lymphocytes? The T lymphocytes we will find surrounding this arteriole, which is called central arteriole, but it is not in the center, off center or eccentric. And surrounding that are the T lymphocytes. They are making peri-artery lymphatic sheath, which can be seen in the other part of this diagram. Let us zoom out. The peri-artery lymphatic sheath here. As we see, there is one central arteriole coming, and this central arteriole, as it enters the lymphatic nodule, it will be off center. This is the central arteriole. And this central arteriole, it was surrounded by some T lymphocytes. These T lymphocytes are still surrounding it when it has entered the lymphatic nodule. And these T lymphocytes, as you can show them here as well, will be forming the peri-artery lymphatic sheath. And what is the importance of such an arrangement? Let us say there was a case of septicemia and in the blood there were antigens, some bacteria. The bacteria will be spilled in the spleen. And since it is surrounded by the T lymphocytes, the first line of defense, cell-mediated immunity, they'll immediately identify and attack the antigen, the bacteria or the protozoa. And later in the lymphoid follicle, we also have the B cells. they'll be producing antibodies. A bit delayed response, the humoral immunity. So these lymphoid follicles will constitute the white pulp. Most of the spleen is the red pulp. We have already discussed red pulp having splenic sinusoids and cords of Billroth. So about 90% is red pulp and about 10% are the scattered lymphoid follicles, in which you also see one component, PALS. And then we have to compare it with the histological specimen, the actual H&E slide. And as we notice, the schematic diagram we have drawn from the actual histological slide. Before we magnify this slide, let us just have an overview. This is one secondary lymphoid organ which is encapsulated. The capsule is sending some of the trabecula inside, the septations, and then we find there are some lymphatic nodules with germinal center. This is the germinal center, which is pale, lightly stained, and in the periphery it is darkly stained. And these splenic nodules, as you see, they are constituting the white pulp of the spleen. Otherwise, they are scattered in all this area, which is called as the red pulp, having the splenic sinusoids and the cords of Billroth. Now, let us magnify the area to describe some more details of this red pulp and the white pulp as well. So here we find the capsule, and which is some collagen fibers sending the septa inside the trabeculae. Now, to differentiate the spleen from the lymph node, you have one distinguishing feature. As we have seen that even the lymph node is encapsulated structure, but in lymph node, there was a large subcapsular sinus filled with the lymph. And such a sinus is not found in the spleen. So though the spleen and lymph node may appear similar, in the lymph node, under the capsule, there's a large subcapsular sinus which is absent in the spleen. Moreover, in the lymph node, we have cortex, paracortex, and medulla. That kind of arrangement is not found in the spleen. In the spleen, what we have is no cortex medulla, but we have got the red pulp and the white pulp. So what do you find in the red pulp? There are two components which we are talking about: the splenic cords of Billroth, and then we have the splenic sinusoids. In the splenic sinusoids, you have the spilled blood, and in the cords of Billroth, you have the reticular fibers and the LMP cells. You know, lymphocytes, the macrophages, and the plasma cells. So that is the red pulp. Two components. What about the white pulp? The white pulp are these lymphatic follicles, which are basically the B lymphocytes, but there are some T lymphocytes also around the central arteriole. So as we see in the white pulp, we have to talk about two components. One is the lymphatic nodule, and number two is the PALS. This lymphatic nodule, as you will notice, will have a pale or lightly stained germinal center. The B lymphocytes, which are undergoing antigenic stimulation, differentiation, getting ready for mounting immune response, and then moving to the periphery, the darker region. There you have this central arteriole, which is eccentric, and around this central arteriole, you have the periartery lymphatic sheath, the T lymphocytes. So basically, white pulp has the B lymphocyte in the nodule and T lymphocyte around the central arteriole. This is the white pulp in the spleen, upon which we have got one MCQ. Let us look at the MCQ now. In this question, we are being asked about the components of the white pulp and the exception we have to find out. So when we talk about the white pulp, we know it is mostly the WBCs, the lymphoid follicles, and eccentrically we have periartery lymphatic sheath as well. So, as we look at the choice number A, it is mentioned PALS, basically the T lymphocytes which are surrounding the eccentric arteriole. Of course, it is the white pulp. Then what about B cells? B cells are actually present in the lymphoid follicles. So, of course, it is the white pulp. These B cells and the T cells, they are the WBCs making the white pulp. And antigen-presenting cells. Antigen-presenting cells are basically, again, monocyte-phagocyte series and a component of WBCs. So, we can consider it under the white pulp. But when it comes to this vascular sinus, which is basically the splenic sinusoids, the splenic sinusoids are not the white pulp, they are the red pulp. In the red pulp, we have two components, one being the splenic sinusoids, and the second, around them, we are having the splenic cords of Billroth. Now, this could be one of the controversial questions, but still, as we find, the better answer for exception is choice number D, because these vascular sinuses or the sinusoid, the splenic sinusoids, along with the splenic cords of Billroth, they must be considered as red red pulp, not the white pulp. The question is on white pulp and the exception should remain as choice number D. And then we move on to the next topic of discussion, the malt, mucosa-associated lymphocyte tissue. Malt is the mucosa-associated lymphoid tissue and must be located in the mucosa. As we know, in the mucosa, we have three components. Histologically, there will be mucosal epithelium. Under that, there will be subepithelial connective tissue, which is called as the lamina propria. And the third component of mucosa is muscularis mucosa. So, what is the location of malt? It is present in the subepithelial connective tissue, that is the lamina propria, that is the location, and it is covered by the mucosal epithelium. These are non-encapsulated structures, and under the example, we have discussed the secondary lymphoid organs like the tonsil. And then we can also talk about the Peyer's patches, which are in the terminal ileum, and then the malt in the vermiform appendix, the large intestine. Now, when we talk about the tonsil, usually it refers to palatine tonsil, but then we know there is a Waldeur's ring surrounding the oral tube and the nasal tube. And there can be various tonsils under the category, like the lingual tonsil on the base of tongue, palatine tonsil, the pharyngeal tonsil adenoid, and then tubal tonsil at the opening of the eustachian tube. So this is the Waldeur's ring, and we would like to see the structure of the tonsils, specifically more that of the palatine tonsil. So when we are talking about the oral tube, respiratory tube, surrounding that is one Waldeur's ring. And in this Waldeur's ring, there are some tonsil, like the pharyngeal tonsil, which is also called as the adenoid, as evident here. Then there is one tubal tonsil at the opening of eustachian tube. And then you can also talk about two more tonsils, one which is on the palate, the palatine tonsil, and one on the base of tongue, the lingual tonsil. So one in the palate and one on the tongue, the palatine tonsil and the lingual tonsil. They are all under the category of malt, mucosa-associated lymphocyte tissue covered by the epithelium. So what will be the epithelium for these two structures? Because they are in the respiratory tube pathway made with the tubal tonsil behind the nasopharynx or the pharyngeal tonsil, the adenoid. They will be lined by respiratory epithelium, pseudo-stratified ciliated columnar epithelium. Whereas if we are talking about the palatine tonsil or the lingual tonsil, since this is the oral cavity, it will be lined by the non-keratinized stratified squamous epithelium. And the diagram which we are magnifying is palatine tonsil. So the epithelium which you will find is non-keratinized stratified squamous epithelium, and in the subepithelial connective tissue, you have these lymphoid follicles, which you can see having a pale area, germinal center, and in the periphery, it is a darker area. In the palatine tonsil, you'll find there are some crypts also, and these tonsillar crypts are all lined by the non-keratinized stratified squamous epithelium. Let us magnify this diagram and then discuss more details. And once again, we find there are a lot of blue dots here. These blue dots are always representing what are called as the lymphocytes, the cells with large nuclei covering almost the entire cytoplasm, the lymphocytes. If it is the lymphatic nodule, like you see here, it is enlarged view of the lymphatic nodule. You can see multiple lymphatic nodules here, along with the germinal center as well. In fact, this is a magnified view of the palatine tonsil. You can see the crypts also, and these crypts are lined by this epithelium. Under the epithelium, there is subepithelial connective tissue in which we have the lymphoid nodules with the pale staining germinal center. In the periphery, there is darker color. So subepithelial connective tissue with lymphoid follicles, all that you can mark here. This is the crypt, the tonsillar crypt, lined by the epithelium, which we have mentioned, is shown here in magnified view, is non-keratinized stratified squamous epithelium, the similar epithelium as we see in the oral cavity. And in the subepithelial connective tissue, we find this is the lymphatic nodule, mostly having the B lymphocytes. You can see the lymphatic nodule in large numbers here in the subepithelial connective tissue. So what is the function of the tonsils? May it be the lingual tonsil, the palatine tonsil, tubal tonsil, or the pharyngeal tonsil, the adenoid. They are the gatekeepers at the entry point. Whatever food we are taking or whatever fumes we are inhaling, if there is any antigen, any bacteria, any foreign component, that will be detected by these lymphoid organs, and they can become enlarged to mount the immune response, like we can have tonsillitis, inflammation and swelling, and those antigens will be then sent towards the lymph node, and there can be associated lymph node enlargement also. So along with tonsillitis, we have the adenitis. In this question, we are being asked about the malt mucosa associated gut-associated lymphocyte tissue. If it is in the gut, we say gut-associated lymphocyte tissue. So since it is mucosa-associated, it will be present in the mucosa. And which part of mucosa? We have learned that it will be in the subepithelial connective tissue. That means it is in the lamina propria. Now, when you say your answer is choice number B, that is the lamina propria or subepithelial connective tissue, what will you do if this option is not present? Then you have a second answer here. The malt can also move into submucosa. So your answer can be A as well. But B is a better answer than A because mostly it is in the lamina propria but can migrate into submucosa. So do you know any example where the malt is moving into submucosa? And one such example is vermiform appendix. In the vermiform appendix, there are so many lymphatic nodules. They are not restricted to the mucosa. They are encroaching upon the submucosa as well. So maybe this question has double answer, but B is a better answer than A. And now we want to look at the vermiform appendix where we could find the malt even extending into submucosa. Keeping your answer as choice number B, let us look at submucosa of vermiform appendix showing the malt. In this diagram, we have taken a section of the vermiform appendix. In the center, we have the lumen, and since it is a gut tube, it will have 4 layers of gut tube: the mucosa, submucosa, muscular externa, and the serosa. Let us mark them here. Number 1, lining the lumen is the mucosa, and under that is the submucosa. Still exterior to that is the muscular externa, and most exterior, which is shown here, is the peritoneal serosa. Now, as such, when you are looking at the lymphoid nodules with the germinal centers, pale staining, periphery dark staining, these lymphatic nodules should be there in the mucosa, the subepithelial connective tissue, lamina propria. But then you notice, due to huge number of lymphoid tissue, it has entered into the submucosa also. So when the malt is extending into submucosa, it is a rare finding and one such rare occasion when you are looking at the vermiform appendix. So here you can see the malt. Basically, the lymphatic nodule has been extending into the submucosa, which itself is a rare finding because mostly the malt must be in the mucosa. So whenever there is an option of choosing the location of malt, the first answer should be mucosa, subepithelial connective tissue, lamina propria. And if that option is not available, then you can go for submucosa because malt can extend into submucosa as well.